So today's topic is going to focus, and I'm very grateful to Samir, NK, and Kapil, all of them, for giving us this platform where we can share, ideas where the rubber meets the road in some of, as I said, the latest and greatest of genomic research, including CRISPR, which stands for Clustered Regularly Interspersed Short Palindromic Repeats, which is a revolutionary genetic editing technology. Today, scientists are able to make live changes to the DNA sequences of living organisms with a high level of accuracy and ease. Think of it as a search and replace tool in your integrated development environment for DNA and gene therapy. And CRISPR is at the forefront of, using what is we call as Cas9. Cas9 is basically a CAS enzyme, which acts as molecular scissors. So remember the search and replace, which I spoke about. The cut part comes through the CAS enzyme, which we also call as a Cas9. And the search part is through the guide RNA, which is called as gRNA, which is allowed capability to match a specific DNA sequence in genome. So it is amazing that today you can go into molecular organisms' DNA. You can harness it to disable genes. You can use it to mutate genes. You can even insert a new DNA sequence. Right? How cool is that, right? So just like we insert codes into algorithms, we insert codes into semiconductor technology. They're actually able to inject these codes using what we also call as vector viruses, which are viruses which are used. Today we work, in a couple of companies in the US where I sit on the board, including IVB, In Vivo Biosystems. We are doing experiments and also some of these AI/ML experiments on zebrafish and sea worms, where we use the organisms' DNA to kind of cut and paste these genomic sequences. And you can mutate a particular disease. You can send in a good virus within quotes, which we call as a vector virus. And gene editing, gene therapy is very, very expensive in India today. So our goal through our company, UBM, along with Dr. Hiranmath Global Enterprises, to make this affordable and accessible, as well as, I can say, scalable through the use of computing power of AI and ML. So when you look at unlocking genomic complexity, there are different parts to this topic. I'll quickly breeze through these slides. We are going to apply some of the concepts of machine learning, large language models, deep vision, and enhanced variant discovery, functional annotation, where we are looking at exponentially large data sets in these genomics. So when you talk of DNA sequences, you're talking of trillions of cells, and every human being's body has trillions of these cells. And imagine the number of DNAs which we have in our body, and how these methods, which bring about these subtle variations, can act as a cure to some of the most complex diseases. And today, this treatment is available, as I said, in the more advanced countries. We are also working with another company called MedLife Therapy in Harvard Medical School with the former Chief Medical Officer, Dr. Bikash Verma, good friend, where he has brought a lot of these gene editing technologies at manufacturing scale to India. So without much ado, let me move to the next important aspect of also quick, I've given a brief, but if you want to scan and stay connected with me, number of companies which our group is invested in, and I'm a fractional CXO on. I'm very excited to be working with molecular genomics labs here in IIT Delhi, in the Bangalore Bio Innovation Center, where again we are applying some of these concepts. I want to apply a disclaimer that none of this information is confidential from the clientele or the invested companies which our family office has invested in. So when you talk about quantum AI, machine learning, deep learning, and genomics, machine learning and genomics basically, as I said, is based on a pattern learning algorithm. You look at large-scale biological data sets, which are DNA sequences. You look at the individual gene expressions. We call it the DNA sequences. What are the clinical outcomes, mutations or unmutations, which we want to do to reverse a particular disease or a disorder? Or also we want to predict the occurrence of a disease. So today you could take a small piece of, a cell of yours. There are companies like this even in UAE where I live, where you can get around 2,500 AED, which is like roughly around 50,000 rupees. You can know what are your allergens, your bodies, what are the things you are allergic to, what are the kind of diseases you are prone to. So all of this is using a lot of deep learning, AI, ML at the back end. And we specifically use neural networks, deep vision, computer vision. And I'll talk about some of these techniques which we are using today for gene function prediction, for enhancing the promoter mapping, and most importantly, the phenotype classification. Where quantum comes, and it is yet to see the scale of adoption, is we also work in IIT Madras, where I visit often. We have access to computing resources of IBM's quantum computer, and we are at very nascent stages here in India, but the potential is huge for preclinical research, drug discovery, invention of newer molecules. Now, moving on to the actual topic, if you look at some of these techniques in genomics, machine learning, as I said, you could look at gene sequences, you could look at predictions, you could look at detecting some of these anomalies or mutations. Deep learning can be used to identify what are some of the preclinical prerequisites, if I can say so, to model, to use, for example, algorithms such as deep C, AlphaFold, where we can actually bring about the latest and greatest ML AI techniques to create some of these, if I can say, predictions around occurrences of diseases and analyzing of the genomic sequences. Moving on, if I move on to specific technologies here, there's a huge rise in the application, especially in one of the companies, IBB, which we are working in the US, where we actually experiment on zebra organism, but you can quickly model a complete disease occurrence in that. And as I said, you could use the CRISPR technology, where you can scan massive amount of genomic data, genomic data sets, to identify newer types of enzymes which can be injected into these cells, where you get higher specificity. So when you do this search and replace in DNA sequencing, as I said, there are two important components. One is the it comes in basically allowing us to accelerate the discovery of newer sequences which can, as I said, unmutate some of these diseases. So as easy as it's sounding on a presentation slide, it's far more difficult in reality to actually go into a DNA sequence, identify the base sequence, and then do what we call as prime editing, where we insert a new, or we delete the existing and we insert a new type. That's why we said it's almost like search and replace of codes or text in an editor. And the precision which AI and ML brings here is you're able to study and analyze massive data sets of these DNA sequences. You're able to also predict the efficacy and the specificity of some of these DNA changes, especially of the base sequences and the prime editors. And what is the effect? If you see the industry effect of this, is you're able to deliver a precision medicine. For example, in the part of the world which we live in, many of us may not know of genetic disorders such as propionica acidemia, which is a multi-organ failure which happens in children, newborns, or children up to the age of 10. People in Asia Pacific, especially if you look at Middle East population also, it's a very rare disorder. And today the only cure which is available is through Mayo Clinic, and they have a fabulous gene editing technology for this. So traditional CRISPR screening, editing technology, all of this takes time. It is manual, it is siloed. It's a very, very robust process which leads a lot of DNA grouping, regrouping, sequencing, and all of that. But AI and ML can kind of accelerate the discovery and development. So currently, as I said, doing some of this micro injection, creating that vector virus, which is like the good virus, which goes in and edits the sequence. The manual selection of these guides, these prime guides, as I referred to, is suboptimal in efficiency, because you may sometimes go off target by targeting the wrong sequences. So it's not that all the changes which you bring in is always going to bring the right outcome. So also we need to, as I said, when we use C. elegans worms and zebrafish, these are model organisms, microorganisms, where you can replicate the complex genetic disorders or the human diseases at a microscopic scale, and you can develop, using AI and ML automation techniques for the discovery of the right sequencing, which can bring about cure for these diseases. So the current workflows, we use it for molecular design, for drug discovery, for screening, multiple molecules. And when you power this with the today we use supercomputers for drug discovery. But with the increasing adoption of quantum computer, which is yet far from reality in terms of mass scale adoption, we'll see that drug discovery for the rarest disease. Let's look at the current COVID outbreak which has happened, right? So the vaccine came out even with the current technologies at a record speed, possible because of some of the technologies we are talking about. But by integrating AI/ML, for example, bringing in the gRNA scoring, which is a core part of the gene editing, or bringing in deep learning where you can predict the kind of phenotype or behavior when you bring about the changes, or looking for target discovery molecules or DNA sequences, right? Or what is the level of toxicity? All of this, the high level of accuracy and speed is brought about in the R&D cycle by this. We can also use this to kind of massage the data around large research papers. These research papers, again, are very voluminous and manual in current research methods. If you have to go through, let's say, 100 papers, and each paper is talking about thousands of DNA sequences, it's going to be a very manually tiring effort. But you can use AI/ML to do the literature mining. You can use especially generative AI, right? You can use it to recognize some of the entities such as the genes, phenotypes. You can also use natural language processing, a large language model, to bring in some insights into newer patents, whether there'll be patent violations, whether there is any competition to what we are already developing, and also create NLP-based chatbots, which can be internal tools which scientists are assisted with. So we can speed in the discovery, we can support the decision making, and come with a lot of real world simulations. So Gen AI is a huge option when it comes to designing these protein molecules, reinventing newer drug designs, coming up with newer protocols, and you're also using GANs for phenotype image augmentation. Lastly, if I look at the quantum computing potential for CRISPR, target discovery, screening, or even some of the long-term molecular optimization, the potentials or the thing is very, very huge. And the roadmap which we right now have when it comes to using AI, ML, NLP, CRISPR phenotype analysis technologies is we're able to deploy some of these machine learning models. We're able to automate. We're able to bring about these changes which are happening at a submolecular level. We are able to accelerate the drug discovery and the preclinical research into this. So we're looking at a roadmap where we develop fully autonomous AI-driven preclinical discovery engine using these LLMs, using some of these deep vision, deep learning algorithms. For the paucity of time, I'll summarize this in the table. When it comes to CRISPR design, using enhanced AI, Gen AI, ML models, we can build in better ML prediction for CRISPR design. For phenotyping, we can use GAN augmentation, deep vision. And for identifying target molecules, compound screening, we can use a lot of AI machine learning. For identifying levels of toxicity, we can use NLP. And for protein design, we can use a lot of simulation based on quantum computing. Now, the other interesting technology is a company we work with called Aridica, which uses PBMC, or peripheral blood mononuclear cells, which are nothing but like your white blood cells, the immune cells which are part of our body, which resists a new disease. So today, centrifuge technology is very common in most of the molecular genomics labs across the world. But what we are building is an intelligent centrifuge system. And again, we are working with some of the institutes of eminence here, where we can automate the isolation of these peripheral blood mononuclear cell, PBMCs, as we call it, the white blood cells specific class. Like, for example, currently I have a viral throat infection. My WBC count would have gone up like crazy to fight the invading virus. So if I'm able to identify what are the right WBC classified cells which are going to fight against a particular kind of disorder, if I'm able to separate it and use it in certain blood transfusion techniques to invent newer technologies for cures, it'll be great. So this microfluid centrifuge technology, which is basically based on sterilization, is largely automated. But integrating AI-driven, automated, scalable isolation technique, where right from taking the blood sample, using the blood sample to identify the various types of, again, using deep learning, computer vision, segmentation of the blood and identifying can lead to faster separation of this, more cost-effective, more repeatable technologies. And you can then bring in quality control, better quality control, which leads to better outcomes. Also, as I said, beyond the WBC classification, cell classification, screening of the actual microscopic images to identify with precision what are the disease-causing viruses and bacteria, and how is it which I can basically improve that is something which I can do. Lastly, build predictive models where I can optimize some of the protocols, I can detect the anomalies, and I can result in better outcomes for the entire centrifuge experiment. Also bringing in a lot of deep learning, machine learning for data analytics, visualization to analyze some of the outcomes from previous experiments, research papers, and discovering newer trends using generative AI, large language models. What are better centrifugation techniques when it comes to this PBMC separation for newer diseases, something which we can look at. And you can look at mining multiple research papers like we saw for the previous example. And also you could use robotics, AI in terms of robotics, where instead of a human arm doing the separation in the centrifuge at some point, you could actually insert a robotic arm, which you see there. And you could use the power of quantum computing to analyze a multivariable parameter-based centrifuge, where you have, again, these WBCs are going to run in millions. They're not something that's running to hundreds or thousands. So it's not like screening or image processing, which we do with few images and train a model, right? So you need to develop precision over time. So the scope for applying AI, ML, and quantum in this. So right from, if I pyramidize this as, right from clinical impact to reproducing this to creating better data insights and using AI, ML, NLP, and quantum computing for automating this to a large extent, this is the way to go, right? So once again, I thank 3AI and all the organizers for this opportunity. And I hope this session is useful. As I said, if you want to get in touch with me, you can always scan the QR code, and you can get in touch with me on my email, and I'm available to explain and share more insights. And I'm very grateful to our collaborators, whether this is Dr. Hiramat Hospitals or Mayo Clinic or Harvard Medical School, where we are actually developing some of these cutting-edge tools. Thank you very much.